Hello, welcome back to another science stream. Hopefully, my mic works. Wait, one moment. Checking. Okay, it works. So, today we are going to discuss GPCR and RTK. But before that, I want to mention that so far, not so far. We recently reached, oh my gosh, the marker. There you go. This one's much better. So we recently hit, whoa, that's fine. 12,000 subscribers, right? Don't mind the fact that it's now 13k. But we recently hit 12,000 subscribers and I was like, you know what? Why not to celebrate do 1200 jumping jacks, okay? And I was like, oh, but that would be crazy if I try it all at once. So why not divide it by like 10? So it's about 120 times 10 sets. Okay, that works. And guess what? We finished set 1, 2, 3, 4, 5, and 6. So right now I'm going to do which set? The 7th set. Right now. Okay? So I'm gonna do the 7th set. Look at this. So far, we've done about, how many is that? If it's half of 1,200, that's already 600. Right, because it's 6 times 120. 6 times 2, 12. 7. Oh my gosh, it's been 720 already actually. Crazy. Yeah, 720. Nope, this is the first 5. So yeah, we're gonna add a new 120 to it right now. One moment. And the biggest proof is the heart rate monitor. Okay, let's go. One moment. All right. Whoa. Okay, here you go. And adjusting the mic. There you go. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30. All right, that one's 30. And a couple more. Whoop whoop, hello Greg, welcome back to the stream. We are at 30 jumping jacks, remember the thing? Until 1200, actually now we're gonna be 11 sets. So I'm at number 30. Okay, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 48, 49, 50. You know, it's kind of hard. Whoa. Wait lang. I don't, is this the pancreas? No. That's the appendix. It has to calm down. 50. One moment. Breathe. Almost there. Okay. I think we're good. Next. 51, 52, 53, 54, 55, 56, 57, 58, 59, 60. Okay, we're at 60. Almost there at 120, you know, halfway. Halfway there. Halfway. Okay. A little bit more. Let's turn on the AC. Okay, let's go. 61, 62, 63, 64, 65, 66, 67, 68, 69, 70, 71, 72, 73, 74, 75, 76, 77, 78, 79, 80. 80. 80 out of 120. Okay, almost there. 80. Alright. And go. 81, 82, 83, 84, 85, 86, 87, 88, 89, 90, 91, 92, 93, 94, 95, 96, 97, 98, 99, 100. Alright. Let's breathe a little bit first. And then, let's decide. I think I'll count in Chinese for the remaining 1 to 20. Okay. And or Japanese. I think I'll count in Japanese. Let's see. Or Chinese. I think Chinese. Hello, Zoro is always lost 247. Welcome to the stream. Oh, before I go and discuss the GPCR and RTK. Oh, by the way, hi, I'm Millennium, Isekai'd past night empress scientist. Sometimes we do science, sometimes Power World, sometimes Pokemon, sometimes studying while using a treadmill because, you know, we have to be both healthy in mind and body. And then aside from that, there is also the heart monitor of the non-streams. And we are actually celebrating 12,000 subscribers using 1,200 jumping jacks. And I was like, I think I have to divide it into a couple of sets and not just do it all in one go. And today we are at set 7. So, and I've finished 100 jumping jacks. Feel free to watch the VOD from the beginning if you wanna hear the hard mode of the first 100. And now I'm gonna do the last 20 using and count using Chinese. Let's go. 1 2 3 4 5 6 7 8 9 10 11 12 13 14 15 16 17 18 19 20. Finished. Okay. So now that, oh my gosh. Okay, it's a little bit mainit, but it's alright. Wait lang, the clip. Where is my clip? We finished 120. Set 7 unlocked. So how many is that so far? 720 plus 120. Wow, we finished 840 jumping jacks. That's crazy. Okay. So, now that we finished the jumping jacks, where's my eraser? We will move on to discussing science. Ciencia. Because, you know, because we need to. And it'll be a very interesting topic, still about cell signaling, but specifically the GPCR and RTK. For those who know, it would be G protein coupled receptor, and what was the R? R something tyrosine kinase. Oh, yeah, I'll reach that in a bit. Wait, I wanna double check. What is RTK? Receptor tyrosine kinases. Receptor tyrosine kinases. Yeah. See? So that one is GPCR and RTK. So now about GPCR. Oh, I can actually move the mic lower so I can see more stuff now. One moment. I think this one will be much better. 1 second. Don't mind me moving the mic. This one will work. Okay, I think that one helps. I can now see. I can see more. Yeah, this one. Is that okay? You can still hear me, right? Yeah. Okay, so now about G protein coupled receptors. You have your, starting with G protein and just the GTP binding proteins, the GPCR and the G proteins themselves. Let's start there. So you have the plasma membrane, a little bit about neurotransmitters, but this is just specifically about the signaling pathway of GPCR. So for this, it would be, okay, you have the extracellular space, and then you have the cytosol, and then you actually have, wait, I don't want the mic to fall. You actually have the ligand bind. Oh no, I can draw it. You have the ligand binding domain. It goes like this first. The ligand binding domain, aka the area where the ligand will attach, is here. Oh, and this is your plasma membrane. And this part is where the G protein will interact. Okay? G protein interacting domain. And from what I remember, your G protein is actually 3 sets. If it's over here, it's gonna be like this. Your alpha is here. This is the GDP, and then there's the beta, and there's the gamma, beta and gamma. So if the ligand binds over here, where? Over here. Oh no, marker. If the ligand binds here, then that induces your, what was it? Your G alpha to activate. Okay, one moment. GTP binding causes the G alpha to dissociate from the beta and gamma, because the G alpha will interact with here, and then overall that would induce, wait, I have a proper flow of it. Yeah, let's say serotonin binds to the GPCR. GPCR will activate the GS, the whole thing, and then adenylyl cyclase. No. Go back a little bit. We are just activating the top part. So yeah, you have something that binds here. Your G alpha dissociates from the beta and gamma because GTP changes into GDP, and then it will activate stuff like the adenylyl cyclase, and after that CAMP and pKa. So yeah, and regarding the specific thing, if your G alpha is the S versus, it was the stimulatory, yeah, if that's the S part, then it will go through the adenylyl cyclase route, and then increase increase CAMP, and then increase pKa. But if your G alpha is the Q type, then it will go into the phospholipase C and then IP3 DAG route. And I will draw that a little bit more in a bit. All right. Now, yes, 7 transmembrane receptors for G proteins. Oh yeah, I am missing some squiggles. 1, 2. Is it kind of like this? It's 1. Okay, it's gonna be like, where is it? Thank you for those reminders. That's really, really good. 1, 2. The second one and the third one. It's kind of how it looks like. So how is it 7? 1, 2, 3, 4, 5, 6, 7. Oh my gosh, it is 7. Looks so pretty. 1, 2, 3, 4, 5, 6, 7. Very awesome. Keep that up. Wait, that one, we will do a dab for that. Beautiful. Thank you very much. 7 transmembrane receptor for G proteins. All right. So again, regarding the specific GPCR, let's say, well, erase this one. Reviewing if it's serotonin. How do you say that? Anyway, if you let's say serotonin, cal, serotonin. If your serotonin binds to the GPCR 1, 2, 3, 4, 5, 6, 7. If it binds here, GPCR at the neuronal membrane, it would activate the G alpha, the S part, and then adenylyl cyclase is activated. This adenylyl cyclase. This is the same enzyme that converts ATP to CAMP to CAMP. Adenosine triphosphate to cyclic monophosphate. Cyclic adenosine monophosphate. What does that look like? Fine. It looks like this. Oxygen, OH, OH, and then let's say you have adenine. Oh, it looks horrendous. Fine, I will. For the G alpha S to CAMP, I want to be able to write that, to draw that properly. Here. Oh, you need space, bro. So let's say this is adenylyl cyclase in the middle. Adenylyl cyclase. Have the arrow. It said from ATP to CAMP, right? To CAMP. Then it will be, I'll draw ATP, fine. I need a lot of space for the phosphates. Okay, I can just draw it this way. This OH, OH, and then your adenine. I trained for this. Wait a second. We can draw this. Nice. Looks ugly, but it's nice. It's okay. It's kenchana. This is the N. N here. And here and here. Line. This one is your NHH. Beautiful. And now it's gonna be line, O, and then phosphate, oxygen, phosphate, oxygen, phosphate, oxygen. Minus. Double bond O minus, double bond O minus, double bond O minus. Beautiful. So that's your ATP. And if we want it to become CAMP, adenylyl cyclase will with using GS, right? GS will happen, so you get adenylyl cyclase. But then if you have GI, then it would stop that. But let's say it's the green route, it's the go route, then it would make your CAMP. Cyclic, take note, cyclic adenosine monophosphate. Big clue, cyclic and mono. So how does it look like? Looks like this. The house is here. OH, another one of your adenosine. There you go. You have adenosine here. And then, this one's the interesting part. You go like O, then you go O, and then go down. Oxygen. Nope. Phosphate. So you have oxygen here. Phosphate. O. The one O. Minus. Beautiful. So that's cyclic AMP. That's CAMP. Okay? And now your CAMP just floats around in the cytoplasm, and at some point it could go, and downstream effects of CAMP could be activating protein kinase A, which I will draw in a little bit. Oh, by the way, there's an inactive form of CAMP, and that is the 5-AMP. Okay, I talked about camp being a very, very important thing to activate PKA. And how does it do that? Okay. You have, I will now erase this, and then I'll draw the big picture again later. So earlier I said we now have camp, right? I'll draw it with a tinier marker so that it will fit in the picture. So you have camp, cyclic AMP. The marker still, yeah, it can still be seen. So you have this, right? And you have OH, and then you have adenine. Oh my gosh. I hate how I don't like that. It looks ugly. Like this. Like this. House. Good. 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 Very nice. N. No. Here. Yes. Yes. Good. 1, 2. Okay. Nice. NH2. And here. And here. Yeah, right? Yes. Okay, good. And then the cyclic part is here. Oxygen here. Go down with the phosphate here. Double bond O. O minus. Beautiful. So that's your 6 cyclic AMP. How does it work? The relationship between cyclic AMP and PKA? Well. Your PKA looks like this. 4 things stuck together. Okay? Because you have the regulatory subunit. Regulatory. And you have the inactive catalytic subunit. Catalytic subunit. Okay? Subunit. And then for this one, it actually, let's see. This one actually would, what's this called? Would detach. So again, let me make sure I'm writing this properly. You have an inactive PKA right now. All right, is an inactive PKA. Inactive PKA. If you have cyclic AMP, aka now this becomes the enzyme. Cyclic AMP, camp, CAMP, binds to the regulatory subunits. Yeah, it binds to the regulatory subunits of the PKA. So let's say camp is yellow, that one. It would bind to, I'll draw it at this end. It would bind here. These are the regulatory subunits, right? With the catalytic subunits that's inactivated. So the regulatory subunits would bind here, like that. And that would lead to the dissociation. Okay, these would break up. And now your catalytic subunit is free to go around and enter the nucleus. Boom, boom. So that's how you go from GPCR until the activation of PKA. Okay, so the next picture I will draw will try to connect them all into one big picture. I do not know if my marker will be able to do that, but I'll try and make them all fit here. That is a challenge. That is a challenge. But let's go. So let's say you have, oh my goodness. I will draw a cell. Let's start with a cell. A cell, this will be a very beautiful circle. Wow, look at that. Oh, there you go. See? That's a circle. Very nice circle. And this is another circle. One more. We can do it. 1 second. And let's go. See? And it didn't connect. There, that's the circle. And now you have your GPCR, right? So 1, 2, 3, 4, 5, 6, 7. 7 transmembrane receptor. So let's label it. Label the important names. GPCR. Okay. So you have a signal molecule that acts as a GEF, a guanidine exchange factor. Let's say you're nerp. Make it blue. This one is a signal molecule. Okay, it would now the binding of it there would lead the GPCR to act as a GEF, a guanidine exchange factor. So what is that? Well, remember how we have our dear alpha subunit here with a GDP, and it has its friend beta and gamma, right? Alright. With this one, the GPCR acting as GEF would lead the GDP to be changed into GTP, right? GDP to GTP, and now it is active. It is active. So once it's active, this one, I'll draw it again. Your alpha subunit can now dissociate from the beta and gamma, and then it will activate the adenylyl cyclase. So another thing in the picture, purple, the adenylyl cyclase, adenylyl cyclase. And what does that do? That's the same thing over here. Okay, it will be the same thing going from ATP to what? To CAMP. Voila. Yes. And if I want to draw the picture for ATP, I will try to make it fit here. We have your like this, and then you have the OH here, you have the OH there, right? Yes. And then you have your adenine, which goes like this, and like this. You have your N here, and N here, and here, and here, and here, and H2 here. And then here you have your wait, no, not yet. It has a CH2. You have your CH2, OP, 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 O. Double bond, double bond, double bond, O minus, O minus, O minus. Your ATP, adenosine triphosphate, will now become CAMP, and that is this one. It will now become a fancy thing. Still OH here, and still your adenine here, and 6 here, and H2. Adenosine triphosphate is really important on cellular responses, even though it came from the mitochondria. Indeed, because ATP is the currency. It is the money. It is the cha-ching of the cell. It's needed everywhere. That's why it's still here. It's still here. It's still here. It's not done. Yeah, you have your oxygen here, your phosphate, O and O minus. So now we have CAMP. What's the picture about CAMP? Well. Oh, I have to note this down because this one's a really fancy thing. I like the GPCR. If you understand the GPCR, it's nice. But if not, then cry. I'ma cry. GPCR. Picture good. Okay, so now we have camp. It's like evolved Pokemon. Indeed, it just becomes worse and worse. So now you have camp. What does camp do? It is the thing which helps because remember you have an inactive pKa because right now it's still stuck. The regulatory subunits are still stuck there on your inactive catalytic subunits, but the camp can then attach to your regulatory subunits this way, like this here, which then leads to your catalytic subunits to being active now. It is now active for, okay, I'm gonna draw it, inactive pKa to active pKa. And then with this active pKa, what happens? It can, some of these can now go inside your nucleus. Voila. Okay, now zooming into what happens inside. Okay. Oh my gosh, my picture says it's at the bottom, so I'm just gonna make it go the long route. Here. I'm gonna draw it this way. Like that, right? That's the nucleus. But I'm gonna make this go in here just so it matches what I drew in the notes. You have a pore, a nuclear pore over here. It's a big pore. There. Just go inside. Yeah, that one's your nuclear pore. That's a gate. Whoops. Whoa, that's a beautiful nucleus. Okay, anyway. One of those would go inside this one. The glowing thing. And then you have... Whoa. I don't like it. We need more space. So I'm just gonna erase it. No. I think we can try to make it fit. Let's see. You have your camp response, inactive camp response binding protein, aka the Krebs, inactive Krebs, which looks like this. This is your Krebs. But then with the pKa, the Krebs, you now have it sticks here. If it's activated, the Krebs, phosphorylated Krebs binds to your Krebs binding protein. Isn't it GPCR affected mood behavior because it's signaling hormones like serotonin? I think so, yes, because an example I had was the serotonin. So yeah. And then you have your inactive Krebs. If it's activated, it becomes phosphorylated, and the phosphorylated Krebs will bind to the Krebs binding protein, CBP, and Krebs CBP will bind to like a target gene and help with transcription. So that's the big picture. In a different page, I will draw the proper Krebs again because it looks too tiny right now. Okay, erase. Krebs binding protein. Yeah, let's just make it a bit bigger. Okay. So this is the pore. Again, this is your nuclear pore. This is the nucleus first. Wow, that's very ugly. One moment. This is the nucleus with the pore, okay? Gate. That's the nuclear pore. Yeah, okay. That's fine. Much better. So, pKa earlier, the activated one already, phosphorylates Krebs. Yeah, I need to reread my biology book right now. Go, you can grab it. that would be nice, and then we would have more notes. Yeah, pKa phosphorylates Krebs. This is your Krebs. This is inactive Krebs. And then if Krebs is phosphorylated with this sparkly pKa, your Krebs now has a phosphate, and then the phosphorylated Krebs would have the, what's this called? Would have the Krebs binding protein attaching to it. CBP. Okay? And now, this CBP will go and find the target gene that it likes. For example, this part, it would attach here. It would assist like this, phosphate and CBP. There you go. And then transcription happens. Booyah! So that's how you go from GPCR to doing an activation of a target gene. So here's the thing. Let me see. There is a slow camp response. So slow camp response would alter the long-term cellular behavior and memory formation in neurons via Krebs it works via Krebs dependent gene expression. It's kind of slow. And some hormonal, this one's a good one. Let me just see. Some hormonal responses are mediated actually by CAMP. So that's gonna be the heart, liver, thyroid, and all of the other stuff. Okay? But I will draw it. One moment. I'm just looking for the right page. Okay. I think I found it. This one works. Here it is. I'm gonna get the new pen. Yeah. So some hormonal responses are mediated by CAMP. And for example, you have the heart, like the adrenaline. Heart adrenaline, if you have higher heart rate and contractility, beta blockers would inhibit the effect by preventing CAMP in the heart, and it reduces the heart rate and blood pressure. So normally adrenaline would increase the heart rate and contractility, but then if you have beta blockers, it stops that. Liver and glucagon will stimulate glycogen breakdown. And then thyroid, like TSH, thyroid stimulating hormone, would regulate. Allow decipher. Net is acting wonky today, on and off Wi-Fi. Oh dear, is the PLDT doing its thing again? No problem. It's gonna work. Or not work. Just pray to the Wi-Fi chan. Do not be shy. But today, I finished the 120 jumping jacks today. So we finished set 7, and I think that would be, how many is that? Was that 7 times 120? 7, 0, 14, 840 actually. Wow, that's a lot. I think that's a big achievement. Oof, finished. Okay. Now, let's do. Oh my goodness. So I literally taped 2 pages of bond paper for this notes. It's signaling via the PIP2 hydrolysis inositol, what? And DAG. Okay. Calm down, notes. Yeah. Okay, this one. Oh, fine. I'll start drawing it. IP3 DAG. This is crazy. Okay. Slowly. 1 second. I have to organize a thing. I think for IP3 DAG, it became longer because of the structures. But it's fine. We'll get through this. Hopefully. The mental instability. Okay. So this is via PI45B2. Alright. And DAG. IP2 DAG. So, how does PLC beta cleave the phosphoinositol into two secondary messengers. Okay, note, the PKC pathway can be achieved by both GPCR and RTK. That is true. First, I had the very big page about it. Where is it? Not this. That's RTK. It's PIP3. That's AKT. There's the crosstalk. Okay. We could go here to this one, the major one first, and then we will do the nitty-gritty details. Yeah, let's make them talk to each other first. Alright, GPCR and RTK together. Let's go. The marker will be tiny. So GPCR and RTK together. So here you have a very, very long membrane. Because why not? I think I'll need a rectangle. Oh, no. We need space. We don't need. Oh, that's fine. GPCR, how many? 1, 2, 3, 4, 5, 6, 7. So now you have a signaling molecule here. This one, you have the two types. You have the G alpha S and G alpha Q, and then the G alpha S would activate the ligand, and then what was it? It would activate it. It would act as Jeff. GPCR would act as Jeff. GDP into GTP, and then once it dissociates, it would activate the adenylyl cyclase, which would interact and which would activate and make your ATP to CAMP. CAMP would then go make from inactivated PKA, activates the PKA, and then after that you have the transcription factors happening. Transcription regulators. Nice. Okay? So now for the G alpha, the Q1, this would go and do the calcium comodulin pathway. Okay? 1 second. Because this one would activate phospholipase C, which then cleaves receptor tyrosine kinases, bind growth factors that regulate cell differentiation, proliferation, survival, metabolism, and migration. Yes, Slay, that is so true. I will be talking about RTK really soon, hopefully in about 5-10 minutes, and we will see that one to the target proteins. Cell differentiation, proliferation, survival. Yeah, that's why I wanted to talk about this one, because the Raf mech irk. But that one's a fun one too. But yeah, for phospholipase C, it will cleave your PIP2, cleaves the PI 4,5P2 into what? Into IP3 and DAG. The inositol triphosphate. And what does this do? Inositol triphosphate diffuses and goes into the cytosol and would release the calcium from the stores, which would then, and the calcium binds to calmodulin, the CAM. And the CAM would activate the CAM kinase, CAMK, which phosphorylates the others, therefore leading to transcription regulators and the target proteins. It would activate that. But here's the plot twist. The DAG would remain at the membrane and activates the PKC. I have to square the important ones. You have PKA, that's a secondary one. You have your CAM kinase. then you have the PKC. Okay? Greetings, Sheriff. Welcome back to the stream where we do GPCR and RTK today. By the way, finished 120 jumping jacks in the first 10 minutes again, so feel free to catch up in the VODs and hear the trouble that was happening. But yeah, so far we are at 840 out of 1200 jumping jacks. Whoop whoop. And because we recently reached 13,000, yay, I will actually add an 11th set, so it's gonna be 20 jumping jacks in the future total. Hopefully we survive and get through those. But yes, so your DAG, your diacylglycerol, would activate the PKC, but before that can even activate the PKC, we need both the calcium and the DAG. But where does that come from? From here, from the released calcium, because of the IP3. So see, we need them all. You need the protein kinase. You need the calcium and the DAG to activate protein kinase C, which then it is the duo for G protein indeed. And PKC would then activate transcription regulators and target proteins. Yeah. So now, we'll now talk about RTK. Okay, so RTK looks fancy like this. This is the shortcut way, but for now, later I will talk about the specifics about it. But right now for RTK, it phosphorylates and then it dimerizes and then phosphorylates itself more, and then you would then pathway PIP2. You would cleave PIP2. There's a pathway where it connects there, over here. But note, this one is PLC gamma. But if it's the G alpha, the Q version, it would be PLC beta. Okay? So they are different PLCs. In the GPCR version, the PLC is a beta, but in the RTK version, the PLC is a gamma. So, but after that one, it's the same. The PIP2 is cleaved. It becomes IP3 and DAG. DAG floats around, activates PKC. But how does it happen? Well, IP3 would activate calcium stores. The calcium also goes there. Calcium stores would activate the CAM, and CAM activates CAM-K, which is the thing. Now, another thing about RTK, it would activate a funny thing called the MAP kinase pathway. Our dear friend. So for the MAP kinase pathway, it would be first, it recruits GERB, the GERB2, aka the SOS. So I will draw these, but just word version. Where is it? Where's my marker? Found it. Your GERB2 will be, where is it? Here. The GERB2, you have an SH2 domain of GERB2. It attaches to RTK, while SH3 domain is where your RAS-Jeff attaches. A picture would be beautiful, but we don't have space right now. I will draw it very, very soon. RAS-Jeff attaches to it. That's different domains, remember? Now, you have the RAS-Jeff, the SOS, and then this is where RAF, Mech, and ERK happens. Mech and ERK. Who are they? RAS is MAP KKK. RAF is... Nope. Not yet. RAS is just RAS. RAS is just RAS. RAS, RAF, Mech, ERK. RAF is the MAP KKK. Mech is MAP KK. ERK is the MAP K. MAP kinase. That's the important one, which would target the proteins and also a little bit the receptors. All right. So again, RTK. RTK would recruit GERB2. GERB2 would have a couple of domains, and what would attach to it? RAS-Jeff. RAS-Jeff activates the RAS. RAS would activate your map KKK, which is the RAF. No, RAS-Jeff would activate RAS, yeah, and then RAS would activate RAF. It's RAS, RAF, Mech, ERK. ERK is the final one. What do these letters mean? The map KKK, it's map kinase, kinase, kinase. The kinase of the kinase of the kinase. Essentially, the last part, the ERK, is important. The map kinase, kinase. The map kinase. Only 1K. Only 1K. And then the final one for RTK is it would activate the PI3 kinase pathway. PI3 would convert your PI45P2 into PIP3. Note, what's up with PIP3? It would activate AKT kinase, but before that, PIP3 actually recruits PDK1, which would then phosphorylate AKT kinase. And voila, target proteins as well. So, that's a very good big picture. And we will do it again. Oh, okay. Why? Because we're crazy. It just helps. Again, one more time. You have this. You have this. You have, yeah, it's you know, it's the things we do to ourselves. GPCR here, 1, 2, 3, 4, 5, 6, 7. GPCR, RTK. Phosphate, 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 phosphate. GPCR, RTK. Alright, so now you have a signal molecule over here. Okay. It would activate as a Jeff. The Jeff would initiate the exchange from the GDP to GTP. Your alpha dissociates with it, and then your adenyl cyclase can then be free. Adenyl cyclase. And adenyl cyclase would then convert your, I will not draw it. I will just write it strong. Adenyl cyclase will convert your ATP to camp. Nice one. And then your camp would go make your pKa be activated. Beautiful. And then you have your transcription factors over here. And that is if the G alpha is the S version. But if it is the G alpha, the Q version, then it goes to a different route. It is the IP3 DAG, right? Yeah, if it's the GQ protein, it goes like this. It activates the PLC what? PLC beta. And which splits the PIP. What's this one? PIP2. I know, big words. Don't worry, we will draw the big words after. PLC into IP3. Split them up, break them up into IP3 DAG. The IP3, the DAG would go and just activate PKC, but how can that do that? Well, it needs the calcium from the IP3 going inside there. Calcium with the stores and it also activates the CAM-modulin, and the CAM-modulin kinase, and that's another square that we need to do. Goes here, transcription factors, and also the proteins, target proteins. You go like this. And then for the RTK route, it could go do that as well, but it has to be PLC. What? PLC? Gamma. And then it also does our dear map kinase, aka GERB2, into RAS-Jeff, which is the SOS, and then RAS, Raf, Mech, Erk, Raf being map, KKK, Mech being map, KK, and Erk being map, K. And then the last one is from, it would do the PI3 kinase pathway, and the PI3 would convert your, what was it? Your something into the PIP3. That's what I know. So who was the thing? It would be PIP2 into PIP3. And then from PIP3, you will actually do the PDK into the AKT. And then the PIP3 can just go directly into AKT. Because it recruits, PIP3 can recruit PDK and then phosphorylate AKT. And then you can go to and make them target proteins. See? Okay, nice. Now that we drew the big picture, I will draw a more zoomed in version of it just so it makes a bit more sense. To watashi. Who is watashi? To moi. To me. Okay. Now we draw it. So this is the IP3 DAG part. GPCR is activated by ligands. Nice. Ligands. Ligand. Okay, we have the... Who do we have? We have the membrane. This is a big membrane. We can make it smaller actually. Like this. I think that one works. Okay. You have your phosphatidyl inositol. Phosphatidyl inositol. That's the PI. And then you go from that into ATP to ADP. You get the PIP PI4P, then ATP GDP, then you get PIP2, and then after that you get IP3 and DAG. Note. Remember, this is the same split happening from into the IP3 DAG. There's just the zoomed in version. Okay, when the DAG will remain in the membrane and activate PKC. How? DAG looks like a fork, like that. So, and your PKC looks like a car, like this, like that. So, your DAG, let me write it. The DAG will go like this here. But it's not yet activated. Why? Because it needs the calcium, remember? And where does the calcium come from? From IP3's something happening. So you have IP3. IP3 floats around the cytosol, and then it would activate some gates. You have your ER here, endoplasmic reticulum. Remember the stream yesterday? So the ER, you have a ton of calciums here. IP3 would attach here. Calcium can then rush out and go here. Lololol. Now look at that. Once that happens, your PKC is now activated. Yeah. Okay, I said earlier PKC is important, but we're not finished. Why? Because the released calciums will also go and bind to calmodulin, the CAM, and the CAM will undergo a very, very big change, which activates the what? The CAM kinase, CAMK. And also it would also do the myosin light chain, MLCK. But that's just an extra info. Yes, you have the cam kinase happening. So that's the zoomed in version for your IP3 DAG. It's happening over there. Now, I talked about the RTK earlier. Okay, I'm gonna do it now. Here it is. RTK. Okay. Fighting. Wait, let me just do a light stick cheer. Whoop whoop. There you go. So, that one works. Now, we have... Where is it? This one now. We have another one that's just the RTK version. Nope. Draw it out. Force yourself. Draw. Okay. You have the two RTKs. I will draw them in a way that kind of looks similar to how it should be drawn. Here. Like this. Okay. You have the RTK. This is the what? This is your tyrosine kinase domain. This is still inactivated. Alright. Now, for this one, let's see. A signal protein binds and comes in and binds the extracellular domain of an inactive RTK. So something happens. It would attach here. And then the phosphates, after that attaches, these two dimerizes, they would like each other and stick to each other. And then the two RTKs are now together. And then they would trans-autophosphorylate, aka each kinase would phosphorylate each other. Wow. And the phosphorylation activity creates docking sites for it. Parking spots, essentially. So I'll just draw it in like a longer form so that we can see it. There's a lot of parking spots here. Here. Now here. So for the parking spots, there is spot number 1, 2, and 3. Oh no, these have to be very phosphorylated. There, so that it would be activated. Now, the first spot would be for the PI3K. Second is for GAP. Third is for PLC gamma. See? So for the PI3K pathway, that's PI3 kinase, that would be for the PI3 AKT pathway. And then the GTPase activating protein is for your RAS, the G and RAS RAFMAT ERK. Then the PLC alpha is for the PIP. So see, you have the PI3. It just goes in reverse order essentially. PLC alpha, PLC gamma, the GAP, and PI3. Yes, the PI3 mTOR. Wait, what was the mTOR pathway again? I'm not sure if I used that term. mTOR pathway. Many effectors. Okay. So now we have, okay, note, many effectors have the SH2 homology domain, and that would specifically be for the phosphorylated tyrosine. And it would continue to activate the RAS GTPases, which are bound by RTK bound GIF. I will now draw the specific thing that's happening in the map kinase. Rawr. Okay. So, it's gonna be here. I have to mechanistic target of rapamycin pathway. Oh, I did not do the rapamycin pathway. But it's alright. Okay, well, one moment. I think I need to drink water. Oh, it's okay. I'll get it later. So, now we have the RTK. doing the map kindness right now. So I'm gonna draw it here. No, we need space. We need space. We need space. So over here, we finished it. We finished phosphorylating, transphosphorylating it, right? Yes, we did. We finished transphosphorylating. Phosphate, 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 phosphate. And then you would have your SH2 over here. SH2 with the SH3 and another SH3 over here. Well, that looks weird. SH3, SH3. Adaptor protein. GERB2 is recruited to the phosphorylated tyrosines on the RTK. This is the GERB2. Voila. Hello, Lendor. Welcome back to the stream. Oh, by the way, update. I finished the... Remember how I was doing about 1200 jumping jacks? We finished 840 already because I did 120 today again. It's like whoop whoop for the 12,000 subscriber celebration. Oh, and recently I think we just reached 13,000, so that's crazy. But yeah, pretty nice. We have more science and Pal World and Pokemon trainers and Pal World catchers. Now for the GERB2, it gets recruited, and the SH2 domain is the one that binds to the phosphorylated part on the activated receptor. Okay? And the SH3 is for the other people. So GERB2 is finished. What does that do? GERB2 would recruit your RasJeff, aka the sauce. RasJeff would look like this. And like this would be RasJeff. So again, right? RTK to GERB2 to RasJeff. And then that's the SOS. RasJeff would then... Where is it? The RasJeff binds... Oh, where is it? Inactive RAS... Wait, GERB2 recruits RasJeff. Yes. RasJeff binds to RAS, a GTP ace. This is your RAS. The RAS is still has GTP right now. That's inactive RAS. GDP. But with RasJeff, it now switches from GDP to GTP. So now your RAS is activated. So again, review RTK on its SH2, and then it would call GERB. GERB attaches to its SH2. The GERB would call in the RasJeff. RasJeff would bind to the RAS. RAS is now activated because it now has GTP. And after that, guess what? It's the fun part. Your RAS will talk to another thing. This is why it's signaling. They're all walkie-talking. RasJeff would talk to... Oh, RAS would talk to RAF. What is RAF? Map KKK and map KKK. RAS activates RAF by directly interacting with it. Now, your RAF is now activated, right? What does it do? It would phosphorylate using ATP into ADP. It would phosphorylate who? RAS, RAF, MEC. It's gonna be MEC. RAS, RAF, MEC, ERK. RAS, RAF, MEC, ERK. MEC would be map KK. And guess what? This is phosphorylated now. Now, MEC activates who? ERK. That would be map K. and boom, phosphorylated. Now ERK would just phosphorylate different things using ATP to ADP, of course. It would activate like protein X or Y or transcription regulatory protein A, depending on gene expression and all of that jazz. Beautiful. Very beautiful. So yeah, that's why we really need the RAS, RAF, MEC, ERK. Okay? And now, you know what I'm going to do? I'm gonna draw it again. Because why not? One more time. Okay, let's do it. So we have the membrane here. Here. And then you have your... Here, you have your RTK. It's... Oh, it looks weird. Don't mind. This is phosphorylated. It would then recruit GERB, which with its SH2, 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 and then it would call in RAS-JEF, and then the RAS-JEF would talk to the RAS, which has GDP right now, but it would activate it RAS into having the GTP, so RAS is now happy. RAS would directly talk to who? To RAF. RAF is gonna be activated now, and then RAS, RAF, MEC, and then ERK. There you go. See? RAF phosphorylates MEC, phosphorylates ERK. Take note, they're phosphorylating because it's a MAP kinase. This is a MAP kinase, and it is a MAP kinase. Because look, the MAP KK, the MAP kinase is a kinase for this, for the MAP kinase. So it's the MAP kinase of it, so it's a kinase of the MAP kinase. We know this is a kinase of the MAP kinase of the kinase of MAP K. That's why it has KKK or KK or K. And this is just a normal MAP K because it would just phosphorylate the other mini stuff like different proteins and etc. So that's how it works. And then, oh no. Now it's a zoom in of PI3. Oh, that's finished. PI3 into DAG and IP3. And I think the one that we did not do is AKT actually. And I'm gonna do that now. So why not? Why not? Okay. For this one, we have AKT pathway. We have another plasma membrane. Your RTK is here. Looks so fancy. Just gets more what's this called? Disembodied every time. Now you have the ligand binds to the receptor, to the RTK. They dimerize, they autophosphorylate, and then you have your activated PI3K kinase, right? Activated PI3K now. It's now phosphorylated. And then you have phosphorylated PIP2. PIP2 becomes PIP3. And what happens to PIP3? You actually could have PDK and AKT here a little bit because your PDK 1 would need the phosphate, your activated PIP3, okay? And the activated PDK1 would then activate your AKT. Oh, here's the mTOR. PDK1 and mTOR in complex 2. Okay, I'm gonna talk a little bit about it. Hello, iTOR. Welcome to the stream. Oh, you're a new one. Yeah, I'm gonna do the introductions then. Hi, I'm Millennium, Isekai'd Mask Knight, Empress Scientist, and here we do Pal World Pokemon. What was it? Jumping Jacks, Treadmill, like Plague Ink, Rebel Ink, Bookworm, I like Pokemon Ruby, Emerald, Sapphire. Anime, Kdrama also. Right now we are in the science season, so you're seeing a science stream. So, but it alternates sometimes between science streams and especially Pal World. So yeah, feel free to tune in and learn more science. Here you go. So now about the AKT, you have what's this called? Oh, did you see a hat pop in when you chatted? I think you might have seen it. Yeah, the bouncies, because you can change your hat if you type exclamation hat. Like that. So at some point it will change one of the hats there because that's one of the bouncies or mine. It's gonna work at some point. Just wait about 5 seconds. And yep, I think there you go. It became a water bucket. That's my new hat. But yeah, that's how it works. So now you have AKT, the PDK1, and the mTOR is gonna be here. mTOR will talk to the other side of your AKT. So now the two sides of AKT is activated, one by PDK1 and one by mTOR. It fully activates your AKT. And then now the activated AKT would dissociate from the membrane. Oh, it's gonna be here. AKT with phosphorylated sides. And then it would talk to the apoptotic pathway, okay? So here you have the bad. What is this? It's called bad. And then there is an inactive apoptosis inhibitory protein. So if your inhibitory protein for apoptosis inactivated, that just means does it, since bad is pro-apoptotic, its inactivation prevents apoptosis. That's true. That's true. So if you phosphorylate the bad, BAD, phosphorylate it. Now the bad is inactivated. Meanwhile, there's the other one who is active apoptosis inhibitory protein. Yeah, because if you have AKT, essentially you split them up and now your inhibitory protein for apoptosis is okay. It works. All right. So that just means if AKT is screwed up, you don't get apoptosis. But if AKT works, then your apoptosis is inhibited. One more time. Wait. I want to say that properly because I think I was like confusing myself. Yeah. So right now, if you have AKT, it would split these up. And then this is now activated. Your apoptosis preventor is now activated. So you inhibit apoptosis, which is good, wait, is it good? Inhibit apoptosis. Yeah, it depends on certain situations. That's why you need to control this properly, because in the case of, let's say, cancer cells, you want them to off themselves, right? But then your normal cells, especially the healthy ones, you want to keep them for a certain period of time. But at some point, you actually need to get rid of them. So it just depends on what you want to do. Right? Yes. Beautiful. So now, as for the questions, so what are the types of cell signaling mechanisms? You have you have paracrine, juxtacrine, autocrine from the review earlier. What are the types of receptors involved in cell signaling? It was RTK and GPCR. Signal transduction pathway. You have a lot. How is G protein activated and deactivated? G protein, it depends on the GPCR. Explain some signaling mechanisms. There are many. GPCR activation of G proteins. That will be GPCR. If your GPCR is activated, actually, you know what? Let's draw the big picture one more time. Because we're crazy. Why not? Okay, it's gonna be here. One second. So you have this, the big C. You have the GPCR 1, 2, 3, 4, 5, 6, 7. GPCR G GPC wow. G one moment. Technical difficulties. Spelling. GPCR. Good. Now you have the RTK here. RTK. Here you go. Looks beautiful. So now GPCR, it's activated. A signaling molecule goes there. Your adenylyl cyclase is activated because the G protein is now, the G alpha S is now separated from your, what's this called? The G, I need a picture. Beta and gamma. Adenylyl cyclase will convert your ATP into CAMP, and then the CAMP will actually convert your inactive pKa's into active pKa, and the pKa can go inside the nucleus and then just phosphorylate some stuff or trigger some things happening, like this one, transcription. Good. Now, if your G alpha is actually the Q version, then it would activate phospholipase C, which type? PLC beta. PLC would stay above there and induce the split of PIP2 into IP3. Oops, the arrow should be like this. IP3 and DAG. DAG will stay in the membrane until, because remember how it looks like this? DAG looks like this. It would go like yeah, and then DAG would activate PKC. There you go. It's not yet activated fully because IP3 still needs to release the calcium and here, calcium from the calcium stores. After that, calcium would also activate CAM, which activates CAM-K, another important secondary molecule, which would help induce transcription, and some would be for the proteins over here, like this. And then now, you have from RTK, this one would be, could activate that also, but it has to be PLC gamma, and then over here it would RTK, and then you have the recursion of GERB2, and your RAS-Jeff is also called. After RAS-Jeff, RAS is called and phosphorylated, and then RAS, RAF, MEC, ERK, and that would be MAP KKK, MAP K, K, and then MAP K. There you go. That's the important part. Go to the proteins and also transcription. Now the last part is the AKT pathway, which is the PI. This is the PIP3, PI3, P1, 3K. Yeah, there you go. Which splits PIP, which becomes from PIP2 and becomes PIP3, and then PIP3 just could go, okay, PDK1 first, PDK1, and then could be AKT now for the apoptotic pathway. If you activate AKT, then you split the bad part, and then you now have an apoptotic inhibitor active. Beautiful. So here's the plot twist. We actually finished the cell signaling section. Whoop whoop. So yeah, I think for the cell cycle, that will be saved for tomorrow. But yes, thank you very much for coming by to this mini, what's this called? Mini cell signaling stream. Yeah. But that is all for today. See you next time.